Today we will talk about the RNA structure, that is ribonucleic acid, the structure of RNA. This RNA is a single-stranded molecule and it is composed of ribonucleotides. It is a long chain of ribonucleotides. So RNA is a single-stranded molecule, and this RNA is composed of ribonucleotides. It is a chain of polyribonucleotides, or we can say that it is composed of thousands and millions of ribonucleotides. So it is a polymer of ribonucleotides. It is a polymer of ribonucleotides. These ribonucleotides, they are the building blocks of the RNA. Thousands and millions of ribonucleotides get joined to each other and form a long chain of RNA. These four types of ribonucleotides, they participate in the synthesis of RNA. That means we can say that four different types of ribonucleotides are the building blocks of RNA. We have adenosine monophosphate, guanosine monophosphate, cytidine monophosphate, and uridine monophosphate. These are building blocks of the building blocks of RNA. So we can say that adenosine monophosphate, guanosine monophosphate, cytidine monophosphate, and uridine monophosphate, they combine in different combinations and result in the formation of RNA. This RNA, as I told you, that it is a single-stranded molecule. It is like this. It is a single-stranded molecule. We can make it like this. This single-stranded molecule, as compared to that of the DNA. Whereas DNA is double-stranded molecule and RNA is single-stranded molecule. Suppose we have this RNA molecule. It is a single-stranded molecule. So this is sugar phosphate backbone, here sugar phosphate backbone, and here we have nitrogenous bases. So nitrogenous bases can be adenine, guanine, cytosine, uracil, adenine, uracil, guanine, cytosine, uracil, cytosine, adenine, uracil, guanine, cytosine. That means combination can be different. So actually this structure like this, we can just, for the sake of convenience or for the sake of simplicity, we can draw the structure like this. For example, we have phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar. So sugar, to the sugar nitrogenous base is attached. This is sugar. For example, adenine is attached, sugar, guanine is attached, cytosine is attached to sugar, or uracil is attached to the sugar. So we can say that this, as I told you, that RNA is a single-stranded molecule. This is a single-stranded, it has only one strand. This is a single-stranded molecule. This RNA is a single-stranded molecule. Now we'll see the basic structure of RNA, that is of ribonucleotides. If we'll take a ribonucleotide, there are three basic components of the nucleotide. We'll draw it like this. For example, we consider it as a nucleotide. This is sugar component. This is nitrogenous base, and this is phosphate group. So a typical nucleotide has got what? It has got sugar component, nitrogenous base, and phosphate group. So three important components are present in nucleotide. we'll discuss these components one by one. First we'll take the sugar. This is sugar. The sugar present in RNA is ribose sugar. It is ribose sugar, C5H10O5. This is ribose sugar. And which type of sugar it is? It is having 5 carbon atoms. So we can say that it is a pentose sugar. It is having 5 carbon atoms. It is having 5 carbon atoms. It is a pentose sugar. It is a pentose sugar. So we can draw the structure like this. It has 5 carbon atoms. This is carbon number first, carbon number second, carbon number third, carbon number fourth, and this is carbon number fifth. So now we have to just satisfy the carbon valency of carbon. Carbon is always tetravalent. It is having 4 valency. This is carbon, hydroxyl group here, hydrogen here. This is hydrogen here, hydroxyl group here. This is hydroxyl group at the carbon third position, hydrogen. This is hydrogen, and this is hydroxyl group at the carbon fifth. Now we will see, this is, we can just calculate. We have got 1, 2, 3, 4, 5 carbon atoms. Then we have got hydrogen 10, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 hydrogen atoms are present there. Then we have got oxygen 5, 1, 2, 3, 4, 5 oxygen atoms, so 5 oxygen atoms. So it is a ribose sugar. It is what is called as ribose sugar, having formula of C5H10O5. There is a basic difference between DNA and RNA, is that DNA is having the deoxyribose sugar. Deoxyribose sugar. What that deoxyribose sugar is? That is, it is in DNA. This oxygen is absent. So sometimes we say that DNA is 2-deoxyribose sugar. That means at the carbon second of the DNA, oxygen has been removed. So we call it 2-deoxyribose sugar. While in case of RNA, this oxygen is present at the carbon second. So oxygen is present in the carbon second. Then we have got, this is carbon first. This carbon first, nitrogenous base is attached to the carbon first of the sugar. Carbon first of this ribose sugar. So this is the site where this nitrogenous base is attached. So we have got this nitrogenous base. This nitrogenous base attached to the carbon first of the sugar. So by means of bond, that is called as N-glycosidic bond. Glycosidic bond. So we have nitrogenous base attached to the sugar at the carbon first by means of a bond, that is called as glycosidic bond or N-glycosidic bond, and represents the nitrogen of the nitrogenous base. Then we have got another thing, that is this carbon 3 of the sugar. Carbon third of the sugar is having the hydroxyl group. So we see hydroxyl group is present here. Hydroxyl group. It is also important when we say 5' end, 3' end. So at the 3' end of the RNA, what is present? Hydroxyl group is present. And now we say that it is having phosphate group. This phosphate group is present at the carbon fifth. So we have this phosphate group. It is present at the carbon fifth of the sugar, and this phosphate group is attached to the carbon fifth of the sugar by means of a bond here, that is called as phosphoester bond. Phosphoester bond. So this is clear that where nitrogen base is attached to the carbon first of the sugar, phosphate group is attached to the carbon fifth of the sugar, and hydroxyl group is present at the carbon third of the sugar. So this is basic ribose sugar in case of this RNA. Then we say that another important component is ribose sugar. We have decided, but then we have got nitrogenous base. This is first component, this is second, then nitrogenous base. In case of RNA, in case of RNA. We have got two types of, in case of RNA, we have got two types of nitrogenous bases. We have got two types of nitrogenous bases, which is called as adenine, purines and pyrimidines. We can say that two types of nitrogenous bases are present in RNA. We have purines and pyrimidines. Two types of nitrogen bases are present in RNA. One is called as purines, adenine and guanine, and pyrimidine, cytosine and uracil. So whereas in case of DNA, we have got what? We have got thymine, and in case of RNA, we have got uracil. This there is a difference between these purines and pyrimidines. We have got, for example, this is purine. This purine, it is a 9-member molecule. We can draw the structure of purine. A typical purine we can take. Typical purine. Purine, it is a 9-member molecule. We can write purine, it's a 9-member molecule. It is a 9-member molecule where we find carbon and nitrogen. How many carbon and nitrogen are present? Carbon is 5 and nitrogen is, how many nitrogen? 4 nitrogen. So we have this is nitrogen here, this is nitrogen here, nitrogen here, this is nitrogen here. So we have 1, 2, 3, 4, 5, 6, 7, 8, 9. So this purine is a 9-member molecule. Now we have to satisfy the valency of carbon. Obviously, a carbon is tetravalent in all respects. So we have, this is nitrogen at 7th position. So this is a typical purine. It is a 9-member molecule having the molecular formula. It is C5H4N4. That means there are 4 nitrogen atoms, 1, 2, 3, 4, and there are 5 carbon atoms in the purine. It is as is depicted from the diagram. It is what purine is a double ring structure. It is a double ring system. It has a double ring structure, and purines are comparatively larger as compared to that of the pyrimidines. So we have two types of purines. One is called as adenine, another is called as guanine. Then we have got this, these are comparatively larger in size. Then we have got pyrimidine. Pyrimidine. This was purine. Now we will take another, that is pyrimidine. This is pyrimidine. Pyrimidine is a 6-member molecule. It is a 6-member molecule. It would be like this. It is a 6-member molecule. We have 2 nitrogen. We have 2 nitrogen. So we have carbon and nitrogen. We have how many carbons are there? 4 and nitrogen 2. That means 6-member molecule. These are 1, 2, 3, 4, 5, 6. 6-member molecule. We will satisfy the valency of carbon. So it is a basic structure of pyrimidine. It is a 6-member molecule, and its molecular formula is, we have C4, H4 and N4. This is the basic structure of the pyrimidine. So this nitrogen at the 9th position in case of purine, and nitrogen at the 1st position in case of pyrimidine. This nitrogen and this nitrogen, they get linked to the sugar, to the carbon first of the sugar by means of an glycosidic bond. That means this N represents the nitrogen. So this nitrogenous base gets attached to the sugar component by means of a glycosidic bond at the carbon first of the sugar. This is a 6-member molecule. It has got 4 carbon atoms and 2 nitrogen atoms. It has got single ring structure. It's a single ring structure. It has got single ring structure, and it are comparatively, these pyrimidines are smaller as compared to that of the pyrimidines. Pyrimidines are smaller as compared to that of the purines. Then we have got another important component. This was regarding the nitrogenous base. We have discussed the nitrogenous base. Then we'll take phosphate component. That means phosphate group. Phosphate group. This is H3PO4. H3PO4. This is third component. H3PO4. This H3PO4, this phosphate group is attached to, we will make it like this. We'll make it like this. It would be POH, OH, OH, and double bond O. So this is a phosphate group. This is a phosphate group which is attached to the carbon fifth of the sugar, and this phosphate group is attached to the carbon fifth of the sugar by means of a bond that is called as phosphoester bond. Phosphoester bond. Now we can understand, in a RNA molecule, we say that one end of the RNA molecule is 5' end, and another end of the RNA molecule is 3' end. What it means? For example, this is our RNA molecule. This is, for example, 5' end. This is 3' end. 3' end. This 5' end and 3' end. What it means? It means that the carbon, the phosphate group, which is present at the carbon fifth of the sugar, is free on this end. For example, this is an RNA molecule. This end is called as 5' end. This is called as 3' end. This 5' end means what? 5' end, and this 5' end, what it means? It means that the phosphate group, which is present at the carbon fifth of the sugar, is free. So it is called as 5' end or 5 prime. And now we have got 3' end. This 3' end, what this means? 3' end, it is called as 3 prime. That means the hydroxyl group which is present at the carbon third of the sugar is free at this end. So here we can see this is the 5' end. That means 5 prime or 5 prime. That means phosphate group here, and at the 3' end we have hydroxyl group here. So at the 3 prime, this is 3 prime. So we can say that on this end, that is 5 prime, phosphate group is free, and at the 3' end, we say that, or 3 prime, we say that hydroxyl group, which is present at the carbon third of the sugar, is free. So this is what is called as 5' end, 3' end. This phosphate present at the carbon fifth of the sugar and hydroxyl group present at the carbon third of the sugar. This we also observe in case of DNA, where we say one strand is in the 5' to 3' direction, another strand is the 3' to 5' direction. That is in case of DNA. But in case of RNA, we have single strand. So we say that this is for example 5' end, 5 prime. It means that phosphate group is present at the carbon fifth of the sugar is free, and this is 3' end or 3 prime. That means hydroxyl group present at the carbon third of the sugar is free at this end. So two ends are there, 5' end and 3' end. This remember that we have got this, we have explained what do you mean 5' end and 3' end of the RNA. Then this represents the sugar phosphate backbone. This is called as sugar phosphate backbone. This is called as sugar. So I have shown it here. This is sugar phosphate backbone. This is sugar phosphate backbone, and this sugar phosphate backbone it is linked by means of what? It has got phosphodiester bond. The sugar phosphate backbone is called as phosphodiester bond. So phosphodiester bond is found in the sugar phosphate backbone. This is called as sugar phosphate backbone. So that means one ribonucleotide is joined to another ribonucleotide by means of a bond that is called as phosphodiester bond. Then nitrogenous base is joined to this sugar phosphate bond. This nitrogen base is joined to this sugar phosphate backbone by means of a bond that is called as glycosidic bond. Then we say that RNA, it is a chain of polyribonucleotides. What it means? That means thousands in a cell, thousands and millions of ribonucleotides get linked by means of a phosphodiester linkage to form a chain of polyribonucleotides. So this was all about the structure of what is 5' end, 3' end. Now what we say that it is a chain of polyribonucleotides. This RNA is a chain of polyribonucleotides. RNA, it is a chain of polyribonucleotides. Chain of polyribonucleotides. Ribonucleotides. That means hundreds and thousands of ribonucleotides. Hundreds and thousands of ribonucleotides are joined by phosphodiester bond to form the RNA. For example, it would be like this: one ribonucleotide. Obviously, this is nitrogenous base. This is phosphate group present. This is sugar component, phosphate group present at the carbon 5th, and hydroxyl group present at the carbon 3rd of the sugar. Now, this one nucleotide is joined to the another nucleotide. This is phosphate of another nucleotide. Then these two ribonucleotides are joined. This is by means of bond that is called as phosphoester bond. Then this is sugar. It is having hydroxyl group at the carbon 3rd and phosphate group at the carbon 5th, and carbon 1st is having nitrogenous base. Then we will make another. These are two ribonucleotides. We can make another ribonucleotide because I told you that RNA is a chain of ribonucleotides. So it is connected to the phosphate. Then this is another ribonucleotide. This is another ribonucleotide. This is another ribonucleotide. This is another ribonucleotide. So we can say that this is hydroxyl group at carbon 3rd. This is nitrogenous base at the carbon 1st, nitrogen base at carbon 1st, nitrogenous base. It can be adenine, guanine, thymine. It can be adenine, guanine, cytosine, uracil. It can be adenine, guanine, cytosine, uracil. This nitrogen base, for example, it is adenine, for example, uracil, cytosine, uracil, guanine. These are nitrogen bases. This is sugar component. This is sugar component. This is sugar component. This is the phosphate group present at the carbon 5th phosphate group. So this sugar phosphate, it forms the backbone. This is called as backbone of the RNA. There we find what type of bond? That is called as phosphodiester bond. Phosphodiester bond. Here in this diagram we can clearly explain what is 5' end and 3' end. That means this is phosphate and this phosphate is present at 5' end, and this hydroxyl group is present at 3' end. That means what this means, 5' 5 prime. We can say that 5 prime means phosphate group at carbon 5 is free. Phosphate group at carbon 5 is free, and hydroxyl group at carbon 3rd is free. If this is one strand, this is one strand, single stranded RNA. That means it has two ends, one end and another end. At one end we have phosphate group. We have phosphate group of the last of the terminal ribonucleotide that is free, but at the but that is present at the carbon 5, and in that strand the hydroxyl group which is present at the carbon third in the last or in the terminal ribonucleotide, that is free. That is why we call this 3' prime, this end we call 3' prime, and this end we call as 5' prime and 3 prime. So this is the basic structure of the RNA. Then RNA, it has been found that this the basic difference between RNA and DNA is that in case of RNA we have ribose sugar, while as in case of DNA we have deoxyribose sugar. That means we have ribose sugar in case of RNA, ribose sugar. Ribose sugar is that type of sugar where we have, first we have, it is a 5 carbon sugar, but at the carbon second there is hydroxyl group. There is hydroxyl group present at the carbon second. While as in case of DNA we have got, in case of DNA there is a sugar that is deoxyribose sugar, where one oxygen is missing at the carbon second. Then another important thing, that is RNA is single-stranded molecule. RNA is single-stranded molecule. That means DNA is double-stranded molecule, RNA is single-stranded molecule. Whereas 5' and 3' exists also in RNA as well as in DNA. Then we have uracil. Uracil in case of RNA. We have 4 types of nitrogen bases in, there are 4 types of nitrogen bases in RNA. We have adenine, guanine, cytosine and uracil. Whereas in case of DNA we have 4 nitrogen bases. We have adenine, guanine, thymine and cytosine. So there is a difference of 1, that is uracil in RNA, and thymine is found in DNA. So this is the main difference between the DNA and RNA. Now this RNA is called as intermediate molecule. This RNA is called as intermediate molecule. So we have studied a structure of this RNA. This RNA is called as intermediate molecule. Why it is called as intermediate molecule? Because of the fact that it carries the information from DNA to proteins. So we can say that RNA is an intermediate molecule. This RNA is an intermediate molecule. Intermediate molecule. It acts as an intermediate between the DNA and proteins. It carries the genetic information from DNA to proteins. It tells the protein the sequence of amino acids. So when RNA is synthesized from DNA, it is called as transcription. When DNA synthesizes RNA, it is called as transcription. And when RNA synthesizes proteins, it is called as translation. It is called as translation. So what do you mean by translation? Translation is the synthesis of proteins. What is transcription? It is the synthesis of RNA. It is the synthesis of RNA from DNA. That is called as transcription. And now what is replication? Replication is the synthesis of DNA, or RNA replication means synthesis of RNA. Then this RNA was found genetic material in most of the plant viruses. In most plant viruses, RNA genetic material in most plant viruses. For example, we have tobacco mosaic virus, we have tomato mosaic virus. These are the plant viruses, phytoviruses, where RNA acts as a genetic material. We use the term riboviruses for them. We use the term retroviruses for the viruses which are having the RNA genetic material. In certain cases, we have also DNA as a genetic material. For example, adenovirus, where DNA is a genetic material. Phage 174, that is also having a DNA as a genetic material. But in riboviruses, RNA acts as a genetic material. There are 2 types of RNA. We have RNA is of 2 types. One is called as genetic RNA or genomic RNA. What is called genetic RNA, genomic RNA, and another is called as non-genetic RNA. Non-genetic RNA. What this what this genomic RNA or genetic RNA means, that it means that where RNA acts as a genetic material, we say that that is called as genetic RNA. When RNA acts as a genetic material, that is called as a genetic RNA. So in genetic RNA, RNA acts as genetic material. For example, it is found in riboviruses, where RNA is a genetic material. We have, for example, HIV virus, human immunodeficiency syndrome virus. We have tobacco mosaic virus. As I told you, we have Ebola virus, where RNA is a genetic material. Another is hepatitis C virus, where RNA is a genetic material. And last but not least, that is coronavirus, where RNA is a genetic material. So these are called as ribovirus or retroviruses, where RNA acts as a genetic material. Then we have got another type, that is called as non-genetic RNA. This in non-genetic RNA, this type of RNA, non-genetic RNA, it is synthesized on DNA template. That means in non-genetic RNA, DNA acts as a genetic material. So it is synthesized on DNA template. It is synthesized on DNA template, and here DNA acts as a genetic material. DNA is genetic material. For example, type of non-genetic RNA is mRNA. We have mRNA, it is a non-genetic RNA. We have tRNA, it is non-genetic RNA. We have ribosomal RNA, it is non-genetic RNA. So this is regarding the what do you mean by genetic RNA, what is meant by the non-genetic RNA. Then we have this RNA has got diversity. Sometimes in certain cases we have single-stranded RNA, we have double-stranded RNA. For example, we have RNA can be single-stranded RNA as well as double-stranded RNA. This single-stranded RNA is found in some viruses. This single-stranded is found in viruses, in majority of the viruses. For example, we have HIV virus, we have this TMV virus, where RNA is single-stranded. That is, this is single-stranded RNA. Now, RNA can be double-stranded. That is in some viruses. For example, it is Rio virus, where RNA is double-stranded. We have wound tumor virus. In wound tumor virus, RNA is double-stranded. Wound tumor virus, RNA is double-stranded. So RNA can be single-stranded, but exceptions exist in case of Rio virus, where RNA is double-stranded, wound tumor virus, where RNA is double-stranded. This was regarding the single-stranded and double-stranded. Then we have, there was a scientist whose name was Hannes Frankel. Hannes Frankel and Etel. They proved that RNA acts as a genetic material. They proved RNA genetic material. They proved RNA genetic material, and they showed that in tobacco mosaic virus, RNA acts as a genetic material. So they proved that RNA acts as a genetic material in tobacco mosaic virus for the first time. This was Hannes Frankel and Etel. Etel means co-workers, other scientists which have worked with the Frankel. Then we have Occhio, this scientist, he for the first time synthesized in vitro synthesis of RNA. What it means? It means that this Occhio synthesized RNA under laboratory conditions. That is called as in vitro synthesis of RNA. In vivo, there is another term that is called in vivo. That means within living cell. So in vitro means outside the cell, outside the living cell. In vitro means outside the living cell, that is under the laboratory condition, while as in vivo means within the living cell, within the living organism. That is called as in vivo. So we have this type of, then we have RNA world hypothesis. RNA world hypothesis. This RNA world hypothesis was given by Carol Voss. It was given by Francis Crick. Then it was important one was, that is Lassell Orgel. These were the scientists, Carol Voss, Francis Crick, and Lassell Orgel. They stated that they studied the RNA world hypothesis. This RNA world hypothesis means that the first molecules that originated on the surface of the earth, they were RNA molecules, and the other biomolecules which are currently in the living world, for example DNA, proteins, and other biomolecules, they have originated from RNA molecules. So RNA molecules act as a precursor for the current biomolecules. So life originated with RNA molecules. Life originated with RNA molecules, and RNA acted as a precursor for the, and RNA, this was a precursor for other biomolecules like proteins, DNA. This was the crux of this RNA world hypothesis. That means this basically RNA has got two remarkable properties. One is that it has RNA, it can store genetic information. That means it can store genetic information. Another is that it can act as a catalyst. That means when RNA acts as a catalyst, it's called as ribozyme. It is called as ribozyme. When RNA has an enzymatic characteristics, enzymatic properties, we call it ribozyme. And RNA also, we know that it stores genetic information. These two remarkable properties of the RNA make it make them to say that it was the RNA molecules which were first originated on the surface of the earth, and from them other biomolecules originated. So RNA acted as a precursor for the other biomolecules like proteins. Actually, we have got an example of this ribozyme. One of the ribozyme is peptidyl transferase. Peptidyl transferase. This peptidyl transferase, it is a ribozyme. That is an RNA enzyme. It is made up of, most of the enzymes are made up of proteins, but this peptidyl transferase, it is made of RNA. Actually, what this peptidyl transferase is, it is simply one of the components of ribosome. Ribosome has got two components. One is ribosomal RNA and ribosomal proteins. This peptidyl transferase is basically a 23S. It is 23S ribosomal RNA found in Lorentz subunit of prokaryotic ribosome. This peptidyl transferase, it is found in prokaryotes as well as eukaryotes, and eukaryotes it is found in 28S ribosomal RNA of Lorentz subunit of eukaryotic ribosome. Now that means this ribosome in case of prokaryotes, in case of eukaryotes, protein synthesizing machines, they contain an enzyme, ribozyme, which is peptidyl transferase, and this peptidyl transferase is a ribozyme which has catalytic properties. And this peptidyl transferase is one of the most important enzyme in case of prokaryotes as well as in eukaryotes. You may remember that this peptidyl transferase, it is important in protein synthesis. That means it helps in the elongation of polypeptide chain. It helps in the formation of peptide bond. It helps in the formation of peptide bond in protein synthesis. That means it helps in the elongation of polypeptide chain. That is one of the most important enzymes in both prokaryotes as well as eukaryotes. So these two properties, that is having the property to store genetic information and having the property of proteins, that means enzymatic properties, catalytic properties, they made to say that it is the RNA which was first originated on the surface of the earth. So we have discussed about the RNA structure, types and RNA hypothesis. Thank you.